Well, first of all, I want to say thank you to everyone for staying through the whole, however many days you've been here so far. And the job that I was given today is to try to think about some of the next steps. What is the broad picture? What are some of the critical areas that we need to be thinking about going forward to really try to move, you know, in a positive direction across a number of areas. And so I'm going to talk about the journey to effective treatments. And this comes from the perspective of not just HC, but my involvement in other rare diseases and kind of, you know, there is a similar strategy across the rare disease spectrum. And there are individual differences between rare diseases, but there's a number of different things, as Dr. Reyna alluded to this morning, that you just have to meet those steps in order to make progress in developing effective therapies. And it involves work from everyone, from the patients to the families to the doctors, to the scientists. Oh, I wasn't live before? Oh, okay. So key milestones in our journey, just to sort of celebrate a little bit and see that compared to many disorders, I mean, 1971 was when the disease was first described. So that's actually relatively recent. And in 1992 was the first familial report of dominant inheritance in an HC family. That's when I was fortunate enough to be at Boston Children's, and I got to see that family. I know a number of other, Dr. Sotero was there at that time and followed that family. And you know, it was such an unusual and challenging disorder that really got many of us focused on thinking about it in more detail and what could we do, because it was epilepsy, but it wasn't epilepsy. It was migraine, but it wasn't migraine. It was dystonia. It was plegia. You know, trying to understand that. In 1995, we first had the consensus for a standardized diagnostic algorithm, and that was led by Anderman, Accardi, and Vigevano from Italy, and involved a host of clinicians primarily, but also some basic scientists thinking about how can we actually decide what is HC, because of course, as you know, it's very complicated. HC has a lot of different aspects to it. And by 1998, there was the establishment of the first International Registry of Biorepository. And again, I was extremely fortunate to come in at the very beginning of that. And that was directly driven by the AHC Foundation. And that's something to be hugely proud about. And I think that that's gone forward since. And again, that should be something we celebrate. In 2004, we reported that ATP1A2 mutations cause an HC phenotype in a Greek kindred. And then, of course, we looked at many, many genes. There were lots of different techniques that were looked at by many different labs across the world. And in 2004, unbeknownst to us, that was when the RDP gene was published as the ATP1A3. So we knew about that ATP1A3. We had looked at ATP1A2. But this is the interesting thing in the era of molecular advances, is that we're seeing across a host of disorders the same gene causing very different neurologic syndromes. And so that's really important to think about going forward. How are we going to look at these syndromes? How are we going to categorize them? How are we going to approach them therapeutically? Are we going to look for unifying themes that we can target across a wider group of patients, which is attractive? Or do we need to focus on individual phenotypes? Because as you know, as Dr. Ozelius described, RDP is at one end of the spectrum, but there's now an expanding group of patients with a number of mutations that cross the entire spectrum, from the infantile severe epileptic phenotype all the way to later onset or even non-penetrant mutations. That's a huge, huge spectrum, but not unusual. Many, many neurologic disorders have a huge spectrum. But when you're thinking about moving to clinical trials, you really do have to strategically think, how are you going to organize your community? How are you going to partner? How are you going to mobilize not just patients with AHC, but perhaps RDP? Do you want to look at the kids with RDP along with the patients with AHC? How are you going to do the studies? How are you going to get the foundations to work together? Finally, after many years, de novo mutations were found to cause the majority of cases of AHC, meaning new sporadic changes. But we still don't know exactly what the mechanism of that, why those hotspots exist. I think, honestly, a lot of that initial phenotyping that was done by the original group of clinicians who had identified this phenotype really drove finding those 3 hotspot mutations because they did such a good job phenotyping. And now that we have the ability to screen this gene routinely, we're finding a much bigger spectrum of disorder, and that's normal. The same thing happened with Rett syndrome. The same thing happens with many other rare disorders. So what are the critical next steps? And I'm just going to give you an overview. What are the critical next clinical steps, and then what are the critical next steps from a basic science perspective? And if you think about the clinical aspects, I mean, we need further ongoing efforts to characterize the full range of phenotypes. We need to understand what are the shared features among patient cohorts, what are the differences, are the triggers the same or not, how are we going to capture and quantify different outcomes in the various populations, and more importantly, thinking about the broader picture, how are we going to design clinical trials based on knowing what patients are out there, how many patients are out there, what age ranges show different phenotypes, is there progression of disease, is it static? And you know from the publications that have happened, the European group published a paper saying that, that the disease was non-progressive. Many of us feel that that's not true, but it's progressive and it is non-progressive in HC because there are long periods of stability, but with superimposed periods of decline or sudden changes in level of function. And so it's both. Yes, it is static, and yes, it is progressive. And it really depends on perhaps the individual triggers and the child and the mutation and all sorts of things that we need to understand a lot better. Second really important clinical step is to expand the resources to help physicians and families achieve access to proactive and standardized care. This area is, the HC population is woefully underserved. We don't have enough multidisciplinary clinical care available to these patients, and families are in extreme distress a lot of the time because they have very scary episodes, all sorts of different types of episodes, epilepsy, dystonia, and they don't know, should they actually go to the ER this time? Should they not go to the emergency room? Do they need to go to the hospital? Do they not? Is it epilepsy? Is it just one of their usual spells? Is it going to turn into something bad? Is it not going to turn into something bad? Do I treat it? Do I not treat it? It just goes on and on, and that's very confusing. And so as a community of clinicians, that we really need to achieve consensus on what are we going to tell families, what support are we going to provide them, what guidelines are we going to give them, and ideally do that the same way, because you can imagine if you get to clinical trials and you don't have standardization of this, the variation in the care will outweigh the benefit from an effective therapy, even if it truly is effective. That's absolutely a fact, and we really need to work on this just as much as we're working on some of the basic scientific aspects going forward. And we need it for both acute symptoms and chronic symptoms. We need chronic management for all sorts of things that are statically acquired over time. They need physical therapy and occupational therapy and educational plans and behavioral management, and what happens when they start vomiting? What about constipation? What about the heart? How often do you assess the heart? What do you do? It can be overwhelming if you have to face each question yourself every time. But it's not overwhelming, and we do this all the time for rare disorders, if you create an expert consensus model where you say, this is the data we have, it's going to change in 5 years, we're going to have to revise it. Let's start with what we know. Let's start where did they get in trouble? What do we do to manage it? What are the guidelines we give the families? And then make it accessible so they don't have to call their doctor, who may be on call or not, and is the only one who knows about HC in that institution, and anyone else you get on call may not know what to tell you when your child's in extreme distress. Following along that closely is the prospective collection of natural history data. And natural history data is a loaded word, right? Because what natural history data are you collecting? But natural history data, as Dr. Reyna mentioned this morning, is so critical for pharmaceutical companies to be convinced. If you don't have good natural history data, and you have not consistently collected a certain number of outcomes, you can't do a clinical trial because you don't really know what would happen anyway. How long should you make the trial? What is the burden of disease in the population? And so that's something, again, we're really still doing quite poorly. We're trying, to get organized to do that. It requires funding, it requires time, it requires a lot of consensus among clinicians, and of course this is a rare disorder, so it requires broad international worldwide collaboration. We also need, as part of that natural history data, what are the baseline existing care and common interventions that are being given. And like many rare disorders, more and more therapeutic things that are not proven are getting added onto these patients' regimens. So many, many patients are on multiple medications, and they're getting new medications for which we have no data about what it does or how it might interact with a new therapy. So for instance, we heard a little bit about Namenda. We've heard about other therapies. What about if we start doing intrathecal baclofen without a study? What if we start doing deep brain stimulation or whatever? That creates a whole new baseline and further varies the care at baseline and makes it even more confusing unless you get a bigger population if there's a lot of variability about what you're doing. So yes, of course, there's always that push between we want to make sure if there's something that looks promising that we want to make sure our child or our adult or whoever in the family has this disorder, we want to be as proactive as possible. But don't think and really push your physician on what really is the data, and could something be bad about this thing? You know, really carefully look at what's the downside, what's the positive, what's the negative. And for many of the psychotropic drugs that we use for behavior, a downside is they cause dystonia and Parkinsonism. That is a well-known side effect. And so I've often wondered and taken these patients on and off because I wonder how much of is the transition to dystonia because we're adding all these medications. Even flunarizine in high doses, which is a very complex drug, piperarazole derivative, has a side effect of dystonia if you give too much of it. So think about that and all the things we're trying to do to manage these kids. Finally, we need to get consensus, and there was a start at this meeting today, and we've thought about this a long time. Matt talked a little bit about this, Louis VLA. We've talked about it at our medical advisory board meeting. We need specific, objective, reliable outcome measures that capture the burden of disease on the patient, on the family, as you know, for futures in clinical trials. What will they be? How will we measure them? How do we know they're better if we've given a possible treatment? What areas are we going to cover? And I'll get a little bit more into that later. What about the critical next steps on the basic side of things? Well, we need to map other genes and pathways that might cause HC and HC-like phenotypes or syndromes. And this is very important in genetics, and we know we learn a lot from understanding. We talked a lot in this meeting about, yes, the ATP, the pump doesn't work, but what are the downstream problems that it creates? Does it lead to cell death because you get, you know, glutamatergic toxicity? And do we need to separately block glutamatergic toxicity? What's it doing to the different neurotransmitters? What's it doing to receptors? What's it doing to the circuits? Are we having cell death? Are we not having cell death in different brain regions where it really might make sense that you're not going to get those cells back with a therapy, but maybe DBS plus a therapy could work, or maybe baclofen plus a therapy, or some of these other treatments. So that's a really important thing, and by doing that continued work, you're going to increase your opportunities to identify additional therapeutic targets. That's really critical. And to understand, again, how those other pathways might be related to whatever ATP1A3 is doing. And I showed you already, we know ATP1A2 can closely mimic, that mutations in ATP can closely mimic, you know, HC in some cases. So, you know, Al George talked a little bit about this and others. We need to develop in vitro assays. Why? Because in vitro means cells in a dish, or some easily measurable assay that can be done quickly. We can use patient-derived cells. We can use non-patient-derived cells that we use CRISPR or something to make a mutation to try to study what's happening in a given cell. We need assays to measure the pump function so that we can sort of get some idea of if we have a promising therapy that we find, is it going to upregulate the pump function? But then we still don't know anything about what happens when you put it in a whole circuit. And the movement disorder, the circuits that control movement are incredibly complex, and they cross many areas. The cerebellum is critical, the basal ganglia, you know, lots of different regions, frontal cortex, et cetera, are important in movement disorders. What about the behavioral regulation, where there may be all different aspects of this syndrome that are caused by different regions of the brain having disrupted ATP1A3. And that's what we need to sort of look at and think independently about different outcomes as different potential targets. Or maybe if we could just replace ATP1A3 and fix the mutation and do something like that, maybe it would affect all of the brain areas, for instance. But many times when you're identifying drugs that might increase the function of the pump or something like that, when it's not working well enough, or decrease if it's working too much, or stabilizing if it's unstable, you know, there's different approaches that you would take in terms of to try to correct those abnormalities. The next thing that people talked about during this meeting is in vivo assays to characterize disease mechanisms. So the mouse models would be an important example of an in vivo assay. So you create a live animal, but it doesn't have to be a mouse model. There's many, the more animal models in my mind for this disease, the better, because different animal models, both vertebrate and invertebrate. So vertebrate would be mouse and other you know, animals with backbones. Invertebrates are things like worms, you know, things like that. So there are a whole host. There's zebrafish models that are great for drug screening. There's C. elegans models, the worm models. There's Drosophila or fly models of disease. And you can use the different models to better dissect different aspects of a disease often. And it sounds like that's not very parsimonious. It's going to take a lot more money if you have to make more models. But the most successful interventions that have the best data are going to be looked at in multiple systems. They're going to have in vitro and in vivo assays. We're going to use patient-derived cells and non-patient-derived cell-based assessments. There's a lot of different things that have to happen in parallel. And, you know, it is expensive, but we're excited since I've started, you know, in 1998. You know, just think of the tremendous progress, and let's celebrate that progress, because it really is opening up this interest to so many more scientists and clinicians. And I think there is a lot of excitement and a lot of work being done. And I do think the strategy of thinking broadly about moving beyond HC or RDP is critical to think about from pharma's perspective. This is talking about mechanisms of dystonia. Dystonia is a very common problem. What if you find a drug that works on this pump, but it also works across a whole spectrum of dystonic disorders? That would be very powerful for pharma to think about. And so that's why one of the things we did at this meeting, Dr. Hosch, Francisca Hosch, was here for a couple days. She works, she's a child neurologist, and one of the issues is let's look in more detail at this BARS ataxia scale and other ataxia scales to see if we can use that as an outcome measure, because ataxia is also a general umbrella. So you go from an individual disease phenotype to a broader phenotype. That has risks and benefits, but you need to kind of explore both areas. And then finally, what about potential genetic modifiers that provide additional therapeutic disease targets? Lori Ozelius brought this up. I was hoping to hear more from Erin during her presentation. Are there other channel genes or other epilepsy genes that really make a difference when the same mutation is present in the HB1A3 gene? Why does one patient have such a greater severity of phenotype than the other? And what are the additional genetic modifiers? And then further, what are the epigenetic? What are the environmental modifiers? There's a whole host of other things we need to think about. So those are the broad overview. That's a lot of steps. And it is a lot. But the exciting thing is we're kind of starting to hit all of them. We need a lot of momentum. We need a lot of resources and time to do it. But we're getting a lot more done. And we are making tremendous progress. So going back to what we initially asked, back in 1995 at that symposium, what is HC? Is it a migraine disorder? Is it a vascular disorder? Is it a seizure disorder? Is it a movement disorder? Is it hemiplegia or dystonia? Is it sporadic or familial? Is it static or progressive? So far, everything's a yes, as far as I can tell. Vascular, you know, there was one, again, publication, remember, looking at blood vessel abnormalities in skin. That's not been followed up on. Everything is proved to be true here, which is really kind of scary, but also shows you we were thinking along the right path. What about a mitochondrial disorder? I mean, we do see secondary defects in mitochondrial metabolism. We saw that early on. This is why we thought about mitochondrial disorders. What about a channelopathy? Well, there is secondary effect on channel function because it's affecting pump function, which affects the gradients, which can affect other channels. So I think this initial summary really pointed us in the right direction, but it took the advanced technology of genomic sequencing and the new platforms to really find that gene was causative for a large majority of cases in it took a lot longer than it needed to take, basically. So again, we're getting to be, we're expanding more and more what are the overlapping phenotypes that present across the spectrum. Terry Sanger mentioned ataxia telangiectasia, cerebellar syndromes. There's a whole number of inherited cerebellar disorders which share a lot of phenotypes in common, including ataxia and dystonia, sometimes paroxysmal dystonia. There's a number of epilepsy syndromes that have overlap, of course. We have patients who have had temporal lobe epilepsy with HC that's clearly evident with hippocampal sclerosis on the MRI. We have patients with complex partial seizures and generalized seizures and partial seizures. There's a whole host of emerging things and overlap in these syndromes. And yes, question.
I'm sorry for stopping you, but your slide is a point that I think might help some people understand from a foundation. I have my foundation hat right now as a board member. So one of the things that I first wondered about, I don't care about CAPOS, I don't care about RDP. My child has AHC and I want to cure now, damn it. So that was my parent hat. And I didn't want to hear about all these other syndromes until I understood how all this research we're talking about gets funded. And for as hard as everyone in this room works to fund AHC research, to get to those phase 4 drug trials and to get the money we're going to need, we're going to have to include other groups and we're going to have to work with other groups. So when you're talking about these other groups that on the surface as parents we want to not care about, we only want to care about AHC, the way to get our kids help through the drug trial process, I think is what you're telling us, is that we're going to have to expand our world so that we can work together to make that transition. Is that correct?
I think what I'm saying is that in the context of, there's different ways to look at different disorders. And in the context of drug development, sometimes you're targeting a specific gene disorder, but many times we're just targeting epilepsy. This is how all these anticonvulsant drugs first got developed, right? You did screening to look at, did it work for epilepsy in very crude models at first? And then you figured out, oh my gosh, yeah, does it work for this kind of epilepsy? Does it work for that? But if you look at all the anticonvulsants out there, they do lots of different things. And we throw them at patients one after another. We try to think that we're using a hypothesis-driven approach. But one of the reasons I'm a researcher and not a pure clinician is it would drive me insane because what we do is so often not data-driven. It doesn't have. And we focus on groups of symptoms. So there's certain things we use for dystonia. There's certain things we use for myoclonus. There's certain things we use for epilepsy. There's certain things we use for subtypes of different epilepsies. There's certain things we use for migraine. And all of that feeds back into, if you think too narrowly and just go for the cure and say, I just want gene therapy right now, you miss the bigger picture. Going to the RDP comment, I learned so much when I figured I could kicked myself in the butt 20 times because we had actually looked at this gene, but and didn't find anything. We discarded as soon as that publication came out. We actually did some screening, but we did it familial cases that didn't turn out to have ATP1A3 in most cases. We had a group of family that were heterogeneous. And so we weren't actually doing the test on the right group of patients initially. So, but what have I learned from RDP? Well, number one, this really, the most predominant shared feature in my mind that is really important in terms of understanding pathophysiologic mechanisms and preventing progression, what is that acute sudden decline? RDP, they're at a certain level, and then there's this major transformation that occurs, and they lose a lot of function at permanent dystonia. We see that in some of our HC kids. All the time we see it. And I'm just going to pick on Lynn Egan, since she's the president, and Mark. I mean, their daughter really took a huge stepwise decline and developed permanent dystonia, really in one fell swoop. And I think that is the relationship between RDP and HC. So yeah, we want to treat the dystonia, and we want to treat the paroxysmal episodes, and we want to treat the cognitive dysfunction, we want to treat the behavioral stuff, but don't we want to prevent more than anything an acute deterioration? where they're at a certain level and they suddenly decline, that's critical for this field, right? So I don't understand it at all. I don't understand why it happens when the same triggers happen. So, Lori.
One thing about RDP that's interesting are the non-manifesting carriers. So there are people who have the mutation who have not undergone that decline, who have overcome their genotype. So if we could understand how did they do that, if we could understand what prevented them from getting the disease, because they're going to have triggers. You know, the trigger is running. Who doesn't run, you know, after a cab or something, right? So they've got to have had triggers in their life, and yet they've overcome. So if we could understand how did they overcome, that could lead us to a treatment for any, you know, all of these.
Right. So exactly, that's exactly the rationale, and that's how we have to think more broadly. Yes, we want a cure for HC, but yes, we also want treatments, and yes, we also want to understand the disease. And we're focusing on truly the most complicated disease I've ever dealt with. It's really a complicated neurologic disease. So we have to keep that in mind, and we have to keep our minds open and our willingness to broadly collaborate open, because we don't want to reinvent the wheel. Ataxia, telangiectasia, they're doing the same kinds of things for rare disease development. Rett syndrome, they're doing the same things, you know, even unrelated disorders. So there's a lot to learn from those other groups, and there's a lot of partnerships that can happen. So going back to the initial diagnostic criteria for classic HC, I'm increasingly seeing a number of patients who get called HC. They do not meet the classic criteria. Even if I pare it down a little bit, they don't meet, because some of them do have MRI abnormalities now. Some of them do have elevated lactate. Some of them do have, you know, they might not have ataxia or cognitive impairment at a certain age. So you wouldn't normally call them that if you just had the clinical criteria. However, and so is it time for revision? Because expanding the spectrum is good to a point for pharma, it's good for families, it's good for fundraising, it's also good for understanding why is the patient with HB1A2 so similar to this patient with HB1A2 mutation is so similar to this patient with an HB1A3 mutation, but these two patients with HB1A3 mutation are completely different from each other. That's really an important point. So don't, I know there's a lot of controversy, I just hear all this stuff on the blogs and this and that. Don't be selfish thinking that it's doing you any good to focus everything on HB1A3, because that's not how the world works. You know, yes, it would be, it's just, there's a lot more to understand here. And in getting the bigger picture, we need to think about that more broadly. And, you know, of course, you want your child who has the D801N or whatever, you know, the natural thing that pharma's going to say, if they were looking narrowly, is say, oh gosh, well, if we're going to do a targeted gene therapy, let's pick one mutation and we can do that one. But that's not how pharma thinks, because number one, that's going to benefit a very small number of patients overall. They're not going to be able to make any money off any drugs, number one, although they have some incentives to develop drugs for rare diseases. There are incentives to do that, especially if whatever they were developing for a rare disease had a broader application, like treatment of dystonia across the spectrum of other diseases. Wow, wouldn't that be great, right? Because we have, so I think there's lots to think about in terms of all of these aspects. So I think that's something we have to do as an international community, but we're already doing it, but just not consistently. So going back to the phenotype, you know, I would say at least 3 children at this meeting I wouldn't have called classic HC. I wouldn't even necessarily call them HC. Many of the patients getting called HC are only being called HC because their mutation is an HC mutation, and they have no paroxysmal events whatsoever. And our research pharmacist in the back has a son with one of us who really presented with dopa-responsive dystonia. Does he have shared features with rapid onset dystonia, Parkinson, and HC? Absolutely. But is he an HC kid? No, he's not. You know, it's just different. So we have to kind of revisit how we're thinking and how we're perceiving things, but also we can't be too narrow, we can't be too broad, especially when you're talking about clinical trial outcomes. So that's really important to think going forward. So there's a number, I mean, again, what are the targets for outcome measure development? I just think it depends on what therapy that you're going for as to what outcomes are going to be important. So if you were targeting the ATP1A3 gene in terms of upregulating the whole thing, maybe you would have to have an outcome for all of these things. But if you were, say, focusing on a treatment that just to focus on the paroxysmal dystonia aspect, you wanted to help find something that would better treat acute paroxysmal dystonic episodes and decrease the frequency and severity of those episodes, you might take a different approach. And you would like to use scales that are already validated in the pediatric population, that have a normal pediatric cohort, normal children, which is extremely hard to get, that have been used in another disease, that actually fit well to identify and provide some sort of quantitative or semi-quantitative measure of how severe a disease burden for a given aspect of this is. And again, you know, behavior, as you just heard from the last presentation, behavior is complicated because there are voluntary aspects to behavioral and there's involuntary aspects. That's really hard to tease out, so you might have to, that might be a little harder. But if you're talking about hemidystonia or full body dystonia, that might be a bit easier. If you're talking about hemiparesis or hemiplegia, that might be easier. But if they're going in and out of episodes all day, this is what we were talking about today in terms of thinking about how would you measure it, how would you test the burden on the family, the caregivers, and the patient. And it's not so easy to figure out how to measure these things. But other groups with pediatric neurologic diseases are using tools that measure exactly this. There's quality of life tools for severely disabled children. There's tools that we use for CP. There's tools being used for ataxia. There's tools being used for dystonia, not very well, which is why better tools that have been with normative data in the population. I'll just tell you one thing. I know you don't want to hear anything about any other disorders, because I can see everyone fall asleep with the SMA thing. But let's look at Duchenne muscular dystrophy. So many promising therapies for Duchenne muscular dystrophy. Huge problem in the field of Duchenne muscular dystrophy. All these promising therapies that look so great. Why? Because the primary outcome they picked was the 6-minute walk test. The 6-minute walk test cannot be tested reliably in a child less than 6 or 7 years old with Duchenne muscular dystrophy. You have to wait till their disease progresses, and then the therapy is designed not to cure the disease, but to just correct it, to turn them into a Becker instead of a Duchenne. So Duchenne has a much more severe phenotype. So you're waiting until they're 7, and then you're starting with an outcome measure where they've already had significant progression of disease, and you're thinking that's going to work, and it hasn't worked. And that's why there's this angst about all these drugs that are not getting approved going forward that should work but aren't, because we didn't pick the right outcome measure. So think about that. This is really critical, and we need every bit of your feedback about this. There was not enough questions about that. You guys need to think about this. How would I measure my kid? How would I know how to measure it? What would be easy? Could all parents do this? Is there an app? Is there something that is measurable? Is it going to be reliable? All of those things. If you're talking about the, for epilepsy, there are lots of tools and logs and things like that, and Kenneth used that in the fludarabine studies initially. It's very burdensome for parents. And so doing a study for a long period of time, if you're going to do it for 6 months, and there might be placebo, and then you get to do drugs for a while, et cetera, you might end up with a year of having to do a log every day. Think about that. So what would you like that log to look like? And what would be reasonable when you're already sleep deprived, you're already up all the time, you're maximally stressed? How could you figure out how to fit that into your day? So again, this is, you can chart these kind of episodes in a number of different ways. In fact, I think everybody could come up in this room, everybody could come with a different way. We have to come together to do it the same way and then test it. If that doesn't work, then we have to do it a different way and test it. If that doesn't work, we have to do it a different way and test it. That all takes time, money, resources, parents crying over logs, playing with their phones, trying different techniques. So it's really important that we think about that. Again, if you look at the persistent features, dystonia, there are some good dystonia scales. There's about 6 different ones. They aren't well validated in the pediatric population. But again, if the dystonia is consistent, as Dr. Sanger said, there are some really good scales to measure that. Same with the ataxia. When they're inconsistent and fluctuating constantly, that becomes a huge barrier. So if you've got a kid, which we saw throughout the meeting, who 1 minute can do the scale much better than the next minute, that's a risky outcome measure unless you can do it in such a consistent way, or say we're only going to do it when these criteria are met. The kid has to be not having a spell by these criteria, and they have to have done it the same time of day. They have to eat breakfast. They have to agree to do it. There's all sorts of things you have to do to stabilize that. Same thing with if you look at coordination. The kids love doing the PEG test, but there's a lot of variability in the normal population. The PEG test gets better and better and better all through like grade school. So again, it's not great test. You can't do it in the youngest patients, but once you get to a certain age, like an adult, great. It's a great test, right? But in kids, it's a moving target. And so could we do it? We're exploring that, right? Because that would be great, but then they might be able to do it in so many seconds the next time, and then they're going to be having more dystonia, and they're going to totally increase the time that they do it the next day. So that's a problem for an outcome measure. Okay. Balance is a very common one, ataxia. And again, this would be so easy to use in many ways were it not for the fact that these kids fluctuate. So really what you have to do is test what we call retest reliability. If you do a group of kids and you bring them one day and you bring them back the next day and you see, you score this, you say, how long does it take them to walk back and forth across 10 meters along this, or can we measure some quantitative measure of how many times they step off the line, or if they can't even do it at all, do we give it a numerical score? There's a zillion ways you could do this. So, and then again, talking about some of the genetic studies, you know, everyone talks of, you know, celebrates the HB1A3 find, but we've been working really hard at finding lots of genes, and we are continuing to find new genes. And these families also have, you know, very similar pictures. So this is a family that, you know, I saw years and years ago. We finally just found the gene, and it's really critical to find those. And there's very much overlap with this gene. How do those two pathways come together with HB1A3? How is it related? No idea. No idea. Just to give you an idea, so Lori sort of did this a little bit, but just to show you when we initially look at the mutations, here's a problem, right? You've got, out of this cohort of 142, you've got 57 with D801N, you've got 38 with E15K, and that accounts for the majority, and then you've got 10 with G947R. What about all these kids and adults with these? And again, how are you going to target broadly? Well, one way when you're thinking about a target drug therapy is you say, okay, you know, there's you cluster these, you say, okay, this group, I'm going to just make up something. So this, you know, third of the mutations actually is because it's affecting a certain function of the pore or of the pump, and so it's doing this. And if only we can do this strategy, we could target all of those. So we're going to bunch those mutations together. We're going to say these 10 are equivalent mutations, and they do a similar thing, and we're going to be able to do them together. And then we have to back that up by saying, you know, do they have sort of a spectrum that overlaps, and does it make sense, whatever. And that's the way you get a bigger cohort, because if we just do one mutation at a time, we're never going to do it. Not going to do it. So we have to be able to figure out the structure-function relationships, the work that Kathryn Sledner's doing. We have to understand the genotype-phenotype relationships, which all the clinicians are doing in conjunction with the geneticists. All of that has to be worked out very carefully. Here's another family that Shoyano is an example of a family that Jula Ashadi identified that has what we now call FIPWE. Is that what we call it? So I just called it atypical HC at first, which there is really no alternating hemiplegia, so that's not appropriate either. This is how you get into, you know, what's in a name. But again, there are some evidence that there might be some inflammatory component in this disease, as people have been talking about episodes of meningitis or encephalitis in some of our patients in the database. They have a, quote, episode of encephalitis. How they diagnosed that a deterioration in their cognitive function, and when they did the spinal tap, they saw cells in the fluid, which often means inflammation in the brain. That's a target, potentially, if we see that, but are we going to actually tap them every single time? Not in most of our HC kids, right? But maybe if they had an unusual episode, or maybe if there's another way to measure blood cytokines or look at a chemical signature in the blood and say, gosh, is there inflammation ramped up? Do we need to do something about that? What would we do in that setting? Would it help to actually treat really bad acute episodes that are persisting with some kind of immunomodulatory or immunomodulatory therapy of some sort? So that's just something to think about. This was again our original family that we reported with ATP1A2 mutations. You know, this was really a frustrating thing that happened with me that shows the benefit of people working together and not duplicating efforts. So this is a family that I was initially was contacted by a wonderful Greek colleague. He said, we really have this interesting family. Would you like to study this family? We studied this family. We found a mutation H2B1A2. I presented it at a conference in Europe, and immediately they said, where do they live? I told them on the island of Mykonos, and 2 weeks after I left, they then went and recollected the family, and the publications came out back to back. That is such a waste of resources and ridiculous. I never want to see that happen again. We have too much to do. There's too much going on. If Duke is studying a thing and I'm doing something else and somebody else is doing something else in Europe, well, let's talk about it and say, you're working on that one, I'll work on this one, you do that, I'll do this. We do have to do that, right? But we don't want to duplicate efforts. We don't want to create separate registries that collect different data that doesn't match with each other. We don't want to do that. But it is really challenging to do this. And every single rare disease I've ever worked on, this is a problem. It's always a problem. It's never not been a problem. It's often a serious problem. I think we're at I think it's a serious problem for this community, a really serious problem that we need to overcome, you know, and we need to work on it. So this was another example of a family that mimicked AHC. This was an Ashkenazi Jewish family I saw with a CACNA1A mutation. And in that particular gene, that is a calcium channel gene in the brain. And that the phenotypes with this, it's a huge gene. And there's a whole range of phenotypes: epilepsy, migraine, seizures of different sorts, like all different kinds of epilepsies, ataxia, episodic ataxia, febrile epilepsy, and there's all kinds of stuff. This is another example of a gene. So again, a lot of the phenotypic, this particular family also had, the girl had, and boy both, would go into these coma-like episodes, very severe, where they wouldn't wake up for a couple days. And that was terrifying to the parents. So, you know, I think again, what does that have to do with anything going on with our patients with AHCF? And again, this is a family still to be, and there's many of these still out here, exome negative or whole genome negative, but we're still trying to figure it out, where a mom has Hemiplegia migraine plus, and then kids are affected, and they have, for all the world, what looks like AHCF, except they have a couple little unusual things, like they get these swellings on their head when they sometimes and they go up and down. We don't know what that is. And 5 to. So, I mean, there are some overlapping patient families out there that may give us an insight into mechanisms with AHC. So, in terms of the genetic spectrum, I've just showed you what are the different causes. The large majority of cases of AHC are H2B1A3. However, there is a small proportion of these mutations in these genes that look like AHC. And again, what can we learn from those populations? What might they tell us about any relationship to our patients that have H2B1A3 mutations? And again, you've seen this a zillion times, but just to point out, because people didn't really show this, they talked about it. So H2B1A2 is a channel gene that is best known for its relationship to hemiplegic migraine, familial hemiplegic migraine, inherited in a dominant fashion, but also in the years since, many de novo mutations are occurring. And it's predominantly expressed in hippocampal neurons, astrocytes, oligodendrocytes. I simplified this. There's a lot more complexity than that. But in mainly supporting cells, but it is expressed in hippocampal neurons. And then HTP1A3 in the neurons of the basal ganglia, hippocampus, cerebellum, and then a ton of other neurons. You know, and Kathy Swedner has taught me it's also involved in these intrinsic fibers in the muscle, the spindles, the muscle spindles that sort of measure length and control reflexes. And again, they are, there's a lot we have to learn from looking at structure-function relationships between those two channels, okay? So, and I'm not going to go over this, you've heard enough about this, but here's where glutamate toxicity comes in the sense that if the pump's not working, maybe you're going to shut down other aspects that are important, nerve growth factors, glutamate, you know, and other neurotransmitter receptor systems. And again, just to mention that these kids with rapid onset dystonia Parkinsonism in childhood have significant overlapping features with our AHC patients in that many of them do tend to have some fluctuation. So it's not like all of them are static. Sometimes they do have some fluctuation. It's just not exactly the full range of things that you see in AHC. And so maybe you could group some of those kids if you were looking at those with static dystonia, for instance, or something. And maybe I'm not sure, but there's things that we have to think about. This is something that I had that Flavia and Yuri, who's working with me, put together, just to mention that not only are there other genetic modifiers, but maybe there's some epigenetic modifications happening, meaning something that permanently alters chromatin when you have a really stressful time. Or maybe there's something that we don't understand that we need to understand. And epigenetic means basically that there are modifications or changes to the actual chromosome so that in a given point in time, the amount of expression of something from a gene changes because of a modification that makes that gene less accessible to transcriptional factors and things like that. So that's a real entity. We know that sometimes that can be a modifier. Sometimes that's different by gender or environmental factors and things like that. And then, I'm not going to go into that in detail, but we talked about the cerebellar role for dystonia. How much does the cerebellum play a role? We've seen that you can see dropout of cells in rapid onset dystonia patients in the cerebellum. We've seen that on our autopsy patients in HC, older patients that have died with HC in adulthood. And so again, that is a unifying feature. We've also been doing parallel the same actual imaging for some of our children, younger children at MGH, so we can follow them across the lifespan. So for those of you who are participating in the Alison Brashear study at Wakefield, we're also trying to share that paradigm. So when we have a patient that comes in with a dystonic crisis, we'll take the opportunity, exact same scanning paradigm, so we can look at those scans in the same way. And one of the patients that we did recently did have some looked like some increased intensity in the dentate area of the cerebellum, which is something they've seen in the RDP patients. So I think we are starting to see something, but the large majority of patients, at least with classic HC at the miler end, don't have MRI changes until later, but some of the mutations like the E851K do have cerebellar atrophy in a higher percentage of patients. But I think that may be something that we just haven't seen yet because we don't prospectively serially follow older patients with HC. I can barely, sometimes they're so sick of looking at me when I come to this meeting, I can't even touch the adults I met 10 years ago, so much less actually get a MRI scan on them. So I think that, there are a lot we have to learn, and we really want to prevent that. Imaging can be a biomarker. If you could say, we know that in a certain percentage of patients with E815K, by age 10, this many are going to have cerebellar atrophy, and we can show volumetric, we can measure volume, and we show that we do a drug, and we don't get that, or dramatically shifts that, but that's a little bit harder study to do over the short term. Okay, so again, briefly mentioning the models. The animal models are great. Every one has a little different phenotype, and there are different ways to elicit the phenotype, and I think they are teaching us a lot more, but calling it an HC or an RDP or something is a little challenging. Because it then gets people in the mindset, oh, that's not my animal model. I think all of these models, whether they were made to study RDP or HC or something else, tell us something. And but they are expensive, and we want to pick the best models. What are the best models for doing assessment of promising therapeutic trials going forward? So what's required to identify genetic or epigenetic modifiers for rare disease? I mean, there are some great models of this: Huntington's disease consortium, Tourette's disease consortium, other worldwide collaborations. I mean, in general, you need more than 1,000 patients or so, except under rare circumstances where you have large families where you can kind of look at variability within the families. But when you've got largely de novo mutations, you need a lot higher numbers. And strategically, we're going to think about how to do that and how we can share samples and figure out how to partner. And we need consistent and agreed upon definitions and criteria for how you're going to say what their phenotype is. And are you going to call it mild, moderate, severe, and how are you going to differentiate between that? And then we're going to need to allow others to duplicate our data. So we're going to have to kind of share and be able to duplicate data to prove things. So closing up with future directions for treatments, we know, I don't have to go over this in detail. There's so many things we need to do, so many different symptoms to try things. We need the outcome measures to do these things. And in the meantime, it's not a bad thing to try things in your child. As long as you have a backup plan, you fully understand the risks, and you have a way to measure whether you think it helped or not. So even if you can come up with your own way in your own household to say, this is where I think they are now, 3-month baseline. Now I'm doing something new. Try not to do 5 things at once. You know, try to be careful, because one of the things that really does trigger these kids is rapid changes in things. Suddenly stopping therapies, switching out therapies, doing 2 new therapies at once, all this kind of stuff. So what about DBS? What about vagal nerve stimulation? What about autonomic system modulators? What about medications that target increased or decreased expression or breakdown of ATP1A3 in cells? What about other ion channel modifiers, epilepsy drugs, whatever? We've never really done, besides the flunarizine study, we've never ever really done great studies, good studies, even placebo-controlled studies. We just haven't done them here. That's really puts us back at the very beginning. So take-home points, I have 2 slides. Am I doing okay, Bill? All right. Critical to fund pilot studies for scientists and clinicians in parallel. They should be collaborative. They should be committed. And they should present projects that align with the objectives of the organization. It's okay if the AHCF foundation is different than, you know, another foundation. You can do complementary things. It doesn't mean that everybody has to do things together. But it would be nice if you didn't exactly duplicate efforts. Like if you hear an idea and then you say, oh, I'm going to do the same thing because we're going to do this and then we're going to do it again. That's nice to replicate data, but when you've got a small cohort of rare disease patients, it's problematic. It doesn't work very well. Disperse funding across critical areas of need. Try to get a consensus on what you think the critical areas of need are. Disperse the funds. The funds are not provided solely to one project or group. You get a group started, you focus on an area, and then you continue to try to get additional funding for other areas that you know need to be developed in parallel if possible. Do not micromanage clinicians and scientists. They're not going to respond well to it. You know, don't try to ask for too much. You have to give creative leeway to allow scientists and academic clinicians to do their work the way they know best. You can ask them to meet certain criteria. You can, you know, but you know, there are ways to do it, and there are ways that alienate physicians and clinicians and scientists. And again, we have to work to do. We need to work synergistically with the other foundations, again, for complementary goals. And then take home too. We need to collaborate and reach out to pharma regularly and often. Ask them to be sponsors. Teach them about our disease. Find out which companies are interested in rare diseases, like BioMarin, like Biogen. There's a whole number of groups that are very interested in rare disease. Seek early engagement with regulatory. Start talking to the FDA now. Say, what outcome measures, this is a really complicated process, what outcome measures would you even, what are you thinking about? Educate them so that they know when you come through with your first proposal that they've really heard about it, they know what you're doing, and they've thought about it. Rank your top priorities and seek competitive applications for things. If you know there's a really important critical issue, put out a call for proposals. Say, we want to do this thing, and we want to fund the best proposal, and this is how much money we have. We know it might not do the whole thing. This is how much, and these are kind of the strings. Try to minimize not too many strings so you can get people to get excited about it. And then aim for cure long term, but be realistic and celebrate the incremental successes. I love this meeting because I get to see the patients enjoying interacting with other kids and adults. I get to see them in an environment that I don't get to see them in the hospital because they hate the hospital. And they're poked and they're prodded and they've been there way too much. And like any kid with a chronic disease, they don't want to be there. And, you know, half the time they're in a quad episode or they have a dystonic crisis. Or, you know, I have to stop. I don't get a good neurologic exam. This is why our notes are crap, right, in the medical notes, because half the time you go to the clinician and like could not examine, you know, aggressive behavior, tried to strangle me, hitting, you know, mother, laying down on the floor, refusing to have blood draw, 7 people to hold child down to have blood draw. We just had a terrible experience in Boston where the parents routinely expect that they know when their daughter gets her port change for vascular access, 7 people need to hold her down. I feel like it's a vet, right? You don't want to do that, but if that's what you have to do to keep your kids safe and you feel support, you've got to do it, right? It doesn't make them partial to the physicians, however. Say that. So acknowledgments, there are so many people, so many organizations, didn't even touch on some of the collaborations with MGA now with Laura Zulis and Allison Bashir and so on, but so many people that I've worked with along the way, and I've just loved to be a part of this process. I'm looking forward to collaborating, working on AHC a lot longer and making some good progress. And, you know, I think that especially the families and patients have been incredible because I don't know how you do it on a daily basis, but you make it work, and you survive, and you come to these meetings, and it's awesome. So thank you.
I don't even know if this is on the map, but are there?
You had to pick a topic that I didn't talk about after all that I threw at you.
So for the common,
I thought I would overwhelm the crowd.
The common genes that you're finding are associated with AHC. Do we have any idea what the population of people is with that mutation that aren't presenting anywhere, anything?
Unless we did whole genome population studies, I mean, we're starting to get, you know, some great databases out there. So, so many of the new cases are being discovered by the exome, the clinical exome. Once we get to genome, we're going to find a lot more. And all of these atypical phenotypes are not coming because a doctor recognized. And I have a couple kids now with H1, H3, and something else, a second mutation in different genes. So it's, you know, there's a lot of, you know, and there's really two mutations. And, you know, I think that until we have, you know, even a million genomes isn't enough. That's the goal for NIH. They're doing the Million Genomes Project right now. But once we have a million, you can start to look at what the frequency of a de novo variant is, but the populations they're collecting don't represent the general population. So until you're doing, and whether that will ever happen, if we all got newborn screening and they did our genome, I think in the US, that's never going to happen. I mean, I think there's too many ethical issues and so on. But honestly, newborn screening for this disorder, if you had effective therapeutic, HC is so bad and presents so early, often in that first 18 months and often in the newborn period and the first few months of life, it would make sense to do newborn screening for that if you could find a cost-effective way to do it. And you can't do that unless you have something to offer parents that really makes a difference. You know, you can't hypothetically say, wouldn't it be great if we did because we would do this or that. That's not how newborn screening works. Yes, Dennis.
Yeah, I have one question, and you made me think of it when you said in the U.S. It's unlikely. What about a long time ago when I went up to Boston Children's with Jimmy? Mm-hmm. There was a Japanese doctor and an Israeli doctor who came there because there were 3 kids with AHC at the same time, and they wanted to see this unusual event. Is Japan doing anything?
Oh,
you know, something where there's like a monoculture or whatever you call it. I mean, they shouldn't have too much genetic variations as in America. I mean, they're not like half Irish, quarter this, eighth that. You know what I mean? Well,
the Japanese have done some of the most beautiful work and the best genotype-phenotype correlation work because they are absolutely wonderful, very meticulous clinicians who are incredibly observant. And the work they did, I think the genetic background in the Japanese population is different as it is in Asians to some degree. But remember, we're mostly much more alike than we are different. But I think that their E815K mutations might look a little worse in their patients, so what they describe than what we see in our cohort in the U.S. And again, we need 1,000 patients to really start to figure that out. And we were commenting that, you know, we do see it across ethnic background, but how many times do we have African-American families at this conference? Not very often. Is that because they don't get diagnosed? Is that because it's less frequent in the African-American population or African population? We found that true in SMA, which is another disorder I work on, which I know you don't want to hear about, but we found it's not in Africans because they have a different haplotype where you get an extra copy backup. And so they're much less likely, and their rate is much lower than other populations. There are differences among populations, and again, that's where modifiers come in. Understanding why a certain population doesn't get something is as important as understanding why they get something, right? The new cholesterol drug, for instance, was found in African Americans with a severe hypercholesterolemic phenotype a novel gene variant was detected, and that's targeted for the cholesterol drug, but the cholesterol drug works on everybody. So I think that's a really important lesson. We're more alike than we're different, and you can learn lessons across that spectrum.
Hi there. Question on, I thought I heard there was a drug out there that I didn't hear talked about the last couple days that has been an experiment. I mentioned it when we met yesterday real quick, but I looked at my paperwork. I don't have it.
Yeah. You mean
for to cure HC, or that's for AHC that is in trials?
You know, no, I mean, there aren't, I think there are sort of mini trials. There's not a well-controlled trial going on for anything. But there are open label studies that people are trying to do, but they're not, it's really mostly like one clinician trying something in a small number of patients and things like that. But yeah, we need to do a lot better than that. But there's
okay. Now our neurologist in Detroit said, "Oh, something will be out in the next year." You know, so over here, like
if there is,
I'm not hearing that here.
Yeah, I don't know about it.
All right, this will be our last question. We're running a little bit behind, and I understand the kids are running around in circles over there. They might be tired.
All right. Hopefully this will be an easy one for you.
Uh-oh, that didn't sound good.
I noticed through your presentation you said, I heard this, or this person said something about something else. And it sounds like there's a minor breakdown in collaboration, or
there always
cohort.
Is in rare disease.
You guys use the term cohort.
Yeah.
What can we as a foundation do to help bridge that gap between different areas of research and helping with
you can use all of the behavioral techniques that Alice shared with you. With all of the clinicians and scientists, just like you do with your kids. I highly advise it.
Can I just add on to that really quick?
Sure.
I'm noticing, I heard Dr. Aaron say it, I heard Dr. Reyna say it, you know, we all need to collaborate, and obviously that's not happening.
It is happening to a large extent, actually,
right.
But there are gaps, like where there's, if someone, I'll just give you an example, if someone wants to lead an effort but they don't seek wide consensus and they do it in a way that other people don't agree with, I don't want to waste my time, right? I mean, it's too hard to do this work. It's too expensive. It's exhausting. It takes a lot of time and money. Why do you want to do something you know is not going to work if you've thought about this for a long time? And so I think, again, communication, it is really hard. But this happens all the time in rare disease, and sometimes it turns out really well. You know, in other areas, sometimes one foundation, I think Parkinson's disease has like 10 foundations just in the U.S. I mean, you know, and they focus on different things, and they're complementary, and they put out different proposals for different areas. But when you're talking about such a rare group of disorders, yeah, if there's not good collaboration or if there's bad behavior, it impacts everybody. It impacts everybody. And so I think, yeah, use Alice's techniques. I think it could work. Put sticky notes.
What do you want to do? We wanted to ask her, of the $20 billion Harvard has, how much can you get? Which we wanted to ask.
Well, I'm working on it. I don't have enough clout there yet.
We don't know what you have to do, but we're not going to question it, right?
Well, Siggy's shared his film with me, so maybe I can get some blue bloods interested. We'll see.
Okay, goodbye to your friends.
All right, thank you, everybody. I think that we have concluded for this evening, and we will see you tomorrow morning at 9 o'clock. Oh, the ice cream social is at 7 o'clock in the room next door. Bye.